When we normally think about behavior, we think about like, what do animals do? What do they produce? But if you start to think about it, you realize that there's a big part to be played by what you don't do. There's the example from the natural world where you're talking about like a predator, that's got its prey in sight, right? And you can watch it and it's clearly preparing to take action, but it's important that it doesn't take that action until the time is right, right? So there's clearly a plan in place, and so there's some kind of drive, right? It's an active process, but there needs to also be a kind of corresponding active process to suppress that drive until the time is right. If you look at the circuitry, if you look at the anatomy of this brain system, one of the most prominent features is that you have these kind of two main pathways. Okay, there are two main feed-forward circuits that course through a number of different brain areas that make up the basal ganglia. They're the so-called direct and indirect pathway. And they're named the direct and indirect pathway because the direct pathway starts in this area that we studied, the striatum, and the projection neurons that initiate that pathway, they send their information directly to output nuclei, thus having kind of an action-promoting function. The indirect pathway, not surprisingly, is called the indirect pathway because neurons that initiate that pathway in the striatum send their information indirectly towards basal ganglia output. So activity in these neurons would be expected, just based on anatomy, to increase inhibition onto targets of the basal ganglia. So there was this picture that they had these opposite roles in guiding behavior. But then when people started to look at the activity of nerve cells in these two pathways, they found that when animals were initiating movements, both of them were active. So this led people to sort of question this idea that they were acting in opposition to each other, one of them promoting movement and the other suppressing movement. So what we did is we said, well, how might you reveal the kind of opposite patterns of modulation in these two pathways? Well, maybe we shouldn't look when animals are acting, but when they're actively suppressing the urge to move. Basically what Bruno did was he trained mice to perform a decision-making task, okay? And it was a task where animals had to judge whether intervals of time were longer or shorter than 1.5 seconds, okay? So they'd be presented with like two beeps separated by variable amounts of time. And if the time between those two beeps was shorter than 1.5 seconds, then the animal would have to make a leftward action in order to get reward. If it was longer than 1.5 seconds, they'd have to go to the right side to get reward, okay? And they learned to do this quite well, right? Now, in this experiment, we added this extra twist, which is that the animal was not allowed to move during that entire interval, okay? So they had to really stay still. So we're studying a timing task, this interval categorization task, but what we're actually doing is we're using it to take experimental control over an internal process, right? An internal motor plan that the animal has to actively suppress. It was striking because we could get an effect on the choices that the animal was making when we inhibited the indirect pathway, the suppressive pathway, on one hemisphere or the other, okay? And we can get an effect on whether the animal was able to suppress the urge to act. But we didn't get a similar effects of inhibiting this action-promoting direct pathway. When we did those manipulations in this particular region of so-called sensory motor striatum. Okay, so this is an area of striatum that's receiving input from lower level sensory and motor areas. And that's when, we started to realize, well, all this suppressive function is tuned, right, for some drive to produce actions, but we're not seeing any function related to action production in this area, so it has to be somewhere else. And that's when Gonzalo started to elaborate his model.
What this model has that's different from other theories of the basal ganglia is that we have multiple loops that are running in parallel, and each one of them have different access to basically what's happening in the environment. But in the end, they're competing for what should be done and what should not be done.
Later, once that model was built, it made this prediction that we should be able to actually inhibit the direct pathway somewhere else and then see an effect on action production.
The reason why we afterwards went into the dorsal medial striatum was more of an educated guess, because we knew that past work has shown that indeed manipulating these more medial circuits that are referred to as more associative circuits can indeed bias decision-making in tasks that are similar to what we were doing.
And so these are areas that instead of just representing the kind of immediate state of the environment, may represent things like, well, what do I think the next state is going to be in the environment, right? They represent kind of more complex knowledge about the world. And so in some ways you can think about what we're showing in the basal ganglia is that it functions kind of like a multi-agent system. The rules, the influences that different circuits learn to have on behavior, they depend on the influences that other circuits are having on behavior, right? They kind of learn from each other, not directly, but through the impact that their influence has on the environment. And this can have relevance for, not only movement disorders, but also sort of more cognitive disorders where, people can't suppress the urge to engage in certain patterns of thought, things like OCD, right? Or Tourette's or something like that. And in fact, some of the same interventions that people use for movement disorders, if you just move them a little bit in terms of the location they're targeting within the basal ganglia, you can treat those kind of more cognitive disorders.